So the blood agar itself contains red blood cells that are either lysed or disturbed through a process that's called hemolysis. Now, there are, I think, 3 types of hemolysis that can be shown by bacteria in this medium, and these include alpha, beta, and gamma. The materials that you will need for the blood agar test include your inoculating loop, your blood agar, your Bacto incinerator, and your unknown bacteria in your slant stock culture. The first thing you're going to do for this experiment is sterilize your inoculating loop. To do that, put that in the Bacto incinerator for 5 to 10 seconds. Once you've sterilized it, allow it to cool off. Once your inoculating loop has cooled off, grab your slant stock culture from the test tube rack. Take the cap off. Flame the test tube for 5 seconds. Then use your sterilized inoculating loop to obtain some of the culture. And then flame the test tube again for 5 seconds. Then put the cap back on. Put it back on the test tube rack. Now, like before in some of the other experiments, we are going to be using the streak method to put our culture that's on our inoculating loop on our blood agar. So, like again, we're going to spread it across the agar, like so. And see how I'm only going in one direction? Put the lid back on. Flame the test tube again. Not the test tube, I mean the inoculating loop for 5 to 10 seconds. Then put it back down on your workbench. Then you're just going to allow your culture to sit for a bit so that the reaction can take place. As always, after you've observed and analyzed your culture, you're going to dispose of this culture in the biohazardous waste container. And after you've done that, you're going to disinfect your workspace. First, you got to move all these materials out of the way. Then you're going to use germicide to actually disinfect the workspace. And then wipe it down using paper towels. And of course, you're going to throw those paper towels away in the biohazardous waste container. And after you've done that, you can disinfect yourself. First, you're going to remove your gloves because they are now contaminated. You're going to put those in the biohazardous waste container as well. And then you're going to go wash your hands. But don't worry, we have one more experiment to do, and that experiment is the motility medium test.